see if I can zoom in a little. There we go, that's better. Continuing chapter 3 on cells. The topics here are membrane function, so how things get in and out, and cell division. There's two main categories for ways things cross the cell membrane. That's passive and active. The difference is based on whether the cells need to use energy to move the substance. In a passive process, the substances are bouncing around and they just move on their own. This includes diffusion. There are two types, simple and facilitated, and then osmosis. Active processes where the cells have to spend energy are active transport and vesicular transport. Diffusion is the process of things just spreading out over time. If there's a bunch of it in one place, it just gradually spreads out until it's more evenly distributed. A difference in how much is where is called a concentration gradient. If you've seen on the roads, a sign like this, it's telling you there's a big hill, that's called a gradient. A concentration gradient is kind of like that. We call it high and low concentration. Low means that there's less of a solute in it. High concentration means there's more of the solute. They're packed more closely together. So, in diffusion, substances move from high concentration to low. We call that moving down the concentration gradient because it's like downhill, or moving with the concentration gradient, as if the gradient is a stream that pushes them that way. Simple diffusion, the particles just move until they fill up the available space to them. To get across a membrane by simple diffusion, it has to be something that isn't blocked by the phospholipids. So something that is lipid soluble, like a steroid hormone, and most gases can get across. So oxygen and carbon dioxide can get in and out of cells easily. And a lot of anesthetics will just go straight in wherever they are. Diffusion of solid particles is what's happening when a substance dissolves and stays in the solution. So we put this little sugar cube here. It gradually breaks up as the sugar molecules spread out. Here they're still more concentrated near where they started, but over time they'll just keep bouncing around until it's equal. That's simple diffusion. For substances that can't just go through the membrane, they have to use facilitated diffusion. They need a transport protein to go through to get across. They're still following their concentration gradient, so going from high concentration to low. It could take them longer to do it if there's a limited number of carriers or channels, which are the types of transport proteins. And that's just due to having, instead of this wide open area where you can go through anywhere, you all have to go through this door. Facilitated diffusion is for small things that are hydrophilic, like ions, small carbohydrates, amino acids. If it's hydrophobic, it can go through without the transport protein. If it's larger, you can't make a transport protein that it can fit through because you'd have too big a gap inside and it wouldn't be able to control other things moving through. So transport proteins come in two varieties. Channels is just an open space. It's specific to certain substances, like this is only for sodium ions. There's a separate one that's only for chloride ions, for example. And the substance can just go through any time, but it's always going to go with its concentration gradients. Carriers are for larger things where, in order to have a space big enough for it, you'd have an open area where you can't control what's going through. So some things that are small enough for facilitated diffusion are too big for an open channel. So instead we have a carrier. A carrier has two positions, open to the outside and open to the inside. It's never open to both sides at once. So it's more like an airlock than an open door. The substance goes in and sits in the carrier, and then it shifts and lets the substance out on the other side. So, something like glucose that's made of 12 large atoms and 12 more hydrogens, which are tiny. If you had just an open channel for that, all kinds of things would be able to sneak through the middle. So instead you have an airlock where glucose gets in, and then we switch and let it through. But it's not sitting there open for anything to get through. So it's kind of like a regular elevator versus a freight elevator. Except that this freight elevator has a key so that only the right substance can make it go. If it didn't have a key, all kinds of things could sit in there and go across with the glucose. So all of your transport proteins are specific for what can go through them. You gotta have the right code to go through the door, even if the door is open. This illustrates a channel versus a carrier. And then here's an animation for facilitated diffusion. Now, this one has no sound. So we've got stuff more concentrated at the top. It's kind of bouncing around. It wants to get to the other side where it's less concentrated, but it can only go through this channel. You might occasionally see the other ones get close to the membrane and then shoot away from it, like they've bounced off the fatty acids. Once this second one goes through, it's equally concentrated, so they're not going to be trying to get one way or the other. They're evenly distributed, so they don't need to diffuse anymore. Questions on facilitated diffusion or simple diffusion? Osmosis is technically diffusion, but the results of it are a little different because what's moving is water. And that's weird because where water is more concentrated, that means there's less solutes. Like if you had pure water versus tea, the pure water is less concentrated in solutes, but it has a higher concentration of water. Water will move when the solutes can't. Solutes want to move and spread themselves out, but if they're not able to cross the barrier, say there are no transport proteins for them, let's give these a full label. water is so tiny that it can sneak through the phospholipid layer even though it's hydrophilic. So when the other solutes can't move and the water can, the water will move to try to make both sides equally concentrated. But water determines the volume of the solution. So if it moves from, here's side B that has only 4 of these solutes to side A that has 8 of them, side A grows and side B shrinks. So osmosis in the body can change the shape of cells. If they take in water, they swell up. If they lose water, they shrivel. We mentioned selective permeability last week. So when a cell is not permeable to some solutes, that can leave the overall concentration unequal on one side or the other of the membrane. The more concentrated area draws the water towards it. We call that osmotic pressure. We'll see the importance of osmotic pressure later in the semester when we get to some things like delivery of nutrients in your cardiovascular system and producing urine in your urinary system.
You ever seen Osmosis Jones?
Yes, but it's been a while. I can't remember enough to know what kind of white blood cell he was supposed to be. There's 5 kinds, and we'll talk about those when we get to cardiovascular too. So, questions on osmosis? Now, there's one more topic that's in the book, doesn't really fit in those active versus passive processes because it's not for crossing a cell membrane. It's for crossing larger membranes like the wall of a blood vessel. It would be passive because cells of that wall don't have to actively spend energy to move substances through, but it's going through pores between the cells rather than going into the cell. What happens in filtration is that you have a physical force that pushes the fluid against the wall or membrane that's there. And that force makes things that are small enough pass through pores. We see this every day in things like straining pasta. The little holes let the water get through and some of the dissolved starch, but not the noodles. Now, brewing your coffee with a drip maker, your filter blocks the grounds, but lets the water and the coffee flavors go through. The bigger the pores, the more things can get through. So if you had a hole in your coffee filter, then the grounds would get through. Filtration in the body is used mostly with blood vessels. The smallest ones are called capillaries, and that's where things are delivered and picked up. So, as the capillaries, or as the blood flows through the capillaries in various tissues of the body, blood pressure pushes it against the walls. There are little gaps between the capillary cells that will let water, small things like ions and glucose through, but block the cells and the proteins that are bigger. So in our bodies, blood pressure is driving filtration. In our examples, gravity was the force. So you couldn't strain pasta on the International Space Station, not unless you had some other kind of force to push the water and the noodles against the filter. Questions on filtration? Our active processes, things where the cells have to spend energy to move substances. Active transport uses transport proteins, specifically carriers, because a channel being open can't control which direction something moves. Uses carriers to push a substance against its concentration gradient. So your facilitated diffusion uses a carrier to move something with its gradient or downhill. Active transport is also using a carrier but it's pushing the substance uphill against the gradient. So to do active transport, you need a carrier protein that can move the substance in the right direction and some energy, usually in the form of ATP, to give it a push, because the substances want to be where they're less concentrated. Why do we want to do this? Move things against their